Hi, I'm Whitney Espich, the CEO of the MIT Alumni Association, and I hope you enjoy this digital production created for alumni and friends like you.
Hi, everybody. Let's get started. Welcome to the MIT Faculty Forum. Thank you all so much for joining us today. My name is Aviva Rutkin, and I'm the data editor at The Conversation, and I'll serve as the moderator for today's presentation, Machine Learning Insights into Antibiotic Lethality. As a reminder, we'll welcome your questions during this chat. If you are one of the alumni who's joining us via Zoom, you can use the Q&A feature, which is found on your toolbar. If you are watching this on YouTube, you can add your questions to the comments field next to the stream. And we also encourage you to tweet using the hashtag #MITBetterWorld. And we'll do our best today to get to as many questions as we can. I am delighted to introduce our featured presenters today, Jason Yang, a research scientist in the lab of James Collins and a member of the Broad Institute, and Allison J. Lopatkin, a postdoctoral fellow in the Institute for Medical Engineering and Science at MIT, as well as the Infectious Disease and Microbiome Group at the Broad Institute as a member of the Collins Lab. Jason received his bachelor's degree in a double major of biomedical engineering and electrical engineering from Johns Hopkins University, and he received his PhD in biomedical engineering from the University of Virginia. As a postdoctoral research scientist in the Collins Lab at the Broad Institute, Yang has been developing systems approaches for understanding mechanisms of antibiotic lethality. He's the recipient of a K99 Pathway to Independence Award from the NIH, and he's interested in developing network modeling and machine learning-based approaches for advancing the practice of precision medicine. Allison completed her PhD training in biomedical engineering at Duke University and her bachelor's degree in applied mathematics at the University of Rochester. Allison's work uses quantitative methods to study the spread of antibiotic resistance within bacterial populations. Her current research interests include modeling the evolutionary dynamics of bacterial communities, leveraging horizontal gene transfer to target pathogens, and examining mechanisms of antibiotic lethality with a focus on the role of metabolism. She'll be joining Barnard College of Columbia University next year as an assistant professor of computational biology. And with that, I'll turn things over to Allison and Jason.
All right, so thank everybody for tuning in. I'm Jason, this is Allison, and we're both postdoctoral research in the Collins Lab at MIT. And we're really happy to have this opportunity to share with you a little bit about some of the work that we've been doing and try to understand how antibiotics kill bacteria.
Okay, great. So I'm going to start out with some high-level review of antibiotics just to make sure we're all on the same page. The discovery and implementation of penicillin really revolutionized modern-day medicine. However, shortly after its introduction, did we start to see the first documented cases of penicillin-resistant organisms in the clinic? And unfortunately, that trend has seemed to maintain since its introduction all the way up till now, where every single antibiotic that we have brought to market has seen the rise of resistant organisms rapidly occur shortly thereafter. And so the rise of antibiotic-resistant pathogens is a growing global threat. The rate at which we can bring antibiotics to market is significantly slower than the rate at which bacteria can acquire resistance to these drugs. And it's actually been predicted that if we don't do something drastic about it, mortality from infectious diseases will skyrocket, potentially reaching 10 million people by the year 2050, becoming the leading global cause of death. And so researchers, around the world are searching and really trying hard to find some type of solution to this. And it turns out that in order to do that, we really need to take a few steps backwards and address or learn more about exactly how antibiotics function so that we can leverage that information and design things in the future that work better. So what do we know about antibiotics? Well, we know that antibiotics target key bacterial processes that are critical for growth. So things like inhibiting the cell wall, things like inhibiting protein synthesis or DNA replication. The antibiotic hits these targets, prevents them from occurring, and eventually the cell dies. Now, it's no surprise then that conventionally, for a very long time, it's been assumed and shown that the faster that a bacterial cell grows, the better the antibiotic is at killing it. So there's a correlation between the growth rate and the killing rate as a function of antibiotics. And that's been shown, and that's true, but more recently, and this is work that has been pioneered by this lab and now validated by many other groups, which has shown that this is not necessarily the whole story. And yes, antibiotics hit their primary target, and that's a huge part of how they function. But following that, the bacterial cell tries to fight back because it has this toxic compound that has now unwittingly been introduced into itself, and it amounts a stress response, and there's a long metabolic demanding process that eventually results in the production of toxic byproducts and ultimately actively contributes to cell death as well. And so this now raises a whole bunch of open questions that our group has been exploring in depth because we were not necessarily aware of all of this before recently. And so one of the very first questions that becomes quite apparent is, because growth and metabolism are so correlated, they're very tightly coupled processes, then perhaps this widely established convention of growth rate as a function of antibiotic killing, well, maybe that's just a proxy for the underlying metabolic state. This is just one example of something that we've been investigating. And so this question, is metabolism a better predictor for antibiotic lethality, is something that is currently in press right now. What we did was we were able to establish a very wide parameter space of all these different environmental conditions, which ultimately amounted to many different growth rates and many different metabolic states, some of which were coupled, some of which weren't. And so we could tease apart growth rate and metabolic state and measure how lethality, or measure how effective the antibiotic was as a function of both of them individually. So you can see here on the left-hand side, across all of these many conditions, when you sort the conditions by growth rate, on the top here, I'm showing you what survival looks like. And you can see very clearly on that left-hand side that there is effectively no correlation between growth rate and survival when decoupled from metabolism. And yet on the right-hand side, that shows that when sorted by the metabolic state, there's a very clear transition between low metabolic state and minimal killing to higher metabolic state and that linear correlation. This is, again, just one example of things that we can start to think about in terms of the role of metabolism and antibiotic lethality. But this is very much the tip of the iceberg. So I'm going to give this to Jason.
Thanks, Allison. So one of the things that Allison and I both have been very interested in has been trying to better understand how it is that metabolism specifically contributes to how antibiotics kill. And as many of you guys may appreciate from things that we were maybe learning in high school chemistry or high school biology, metabolism is really complex. So in the cell, in every cell, in every organism, there are several different processes that are involved in making each of the building blocks that are necessary for any cell to replicate. And in most cases, when we talk about metabolism, we oftentimes are very specifically talking about central metabolism. So those are things like the TCA cycle or a Krebs cycle. And that's shown in the center of this map, in blue with that giant circle in the middle. But as you can see from this diagram, there are many processes that are important for cell division, and these all share substrates and crosstalk with each other, which from a systems perspective actually makes it really difficult to tackle and investigate. And so we were challenged with this question of asking ourselves, how can we better understand biology, in this complicated context? And I would say today there's really predominantly there's been two main approaches that biologists have used recently to discover new mechanisms. One of these is something that we can consider to be a more informatics-driven approach. And so very commonly today we have technology that allows us to do very high throughput or large type of experiments. And these include things like chemical screens or genetic screens. And then here and depicted on the left-hand side, oftentimes what we do is we perform a screen and then subjectively select some criteria by which will determine which of those perturbations were important. And some of those criteria might be things like a certain p-value, a certain effect size. And then what we hope to do in an informatics-driven approach is to take those hits, those pieces that we think are important for the biology, to drive some sort of downstream bioinformatic analyses that we hope can lead us to testable hypotheses. But one of the challenges of this approach is that oftentimes heuristically, the number of hits that you might get from any sort of screen is in the 1 to 3% range. And often, consequentially, often that's too few to really statistically power the sort of downstream analysis that you want to do later. And so consequently, the types of results that come out aren't always experimentally actionable. Alternatively, as I'm sure most of our viewers know today or are aware of today, there's excitement around a class of approaches that are more data-driven. And so these are things like machine learning-based, approaches like deep learning and another sort of new techniques. And the fundamental principle here with these data-driven approaches is that we may begin with some set of biological data, maybe, for instance, biological features that represent the data from our initial screens. And we have with those corresponding phenotype data. So for instance, measurements about how well an antibiotic is killing. And what we want to do is use a computer to determine some sort of transfer function linking the initial biological feature data to our phenotypic data. And while computers are really good at now defining really, really strong high fidelity associations, one of the fundamental barriers here is that many times those associations, those transfer functions are opaque. We often call them black box kind of approaches because the members or those transfer functions themselves can't be mapped to something in biology. So we can't actually mechanistically interpret how those associations are being made. And so to address both these challenges and to kind of further our fundamental understanding of how it is that antibiotics are killing, we recently developed and described an approach that we call white box machine learning, where instead of using machine learning to train a predictive model based solely on the raw data that's in your biological features and your output data, what we're going to do first is use network models to interpret the larger biological feature data into a representative state that already has some sort of mechanistic understanding. And then what we do is we use those interpreted and simulated network states as our machine learning inputs so that the computer is selecting different features that have already been interpreted to explain to us how it is that, in this case, antibiotics are working. And more specifically, what we did in this project, which also recently was published, what we did was we first performed on the very left-hand side a biochemical screen where we took E. coli cells and we treated them with different antibiotics as well as different metabolites that we know participate in E. coli metabolism. And this gave us an empirical input-output relationship between different types of perturbations of metabolism and their effects on antibiotic killing. Then what we did was we used network models of E. coli metabolism to simulate each one of our different experimental conditions. And so this gave us a large matrix of simulated network states corresponding to, in this case, each of our 200 different experimental conditions. And then we took the combination of this empirical antibiotic efficacy data, or antibiotic lethality data, and our simulated metabolic network states together as our machine learning inputs to ask the computer to tell us what reactions can describe or predict antibiotic killing, and what pathways or what processes are they involved in. And this to us became, we view this as an engine that enables us to very rapidly identify mechanisms that can be experimentally actionable and validated. And at a high level, this overall approach addresses the two fundamental limitations that I was describing about on the previous slide, namely that one of the advantages it has over an informatics-driven approach is that we actually get to, because we're not thresholding by some subjective criteria for what data to include and exclude, we actually get to use all of the data that we measure, including our negative data. That actually allows us to enrich and expand the biological information content that is available in our raw data, in our raw screen. And then second, in relation to the more contemporary, purely data-driven approaches, here you can interpret what we're doing here is using the network models as a representation of our existing knowledge bases and basically giving our analysis additional prior knowledge by which we can interpret the more raw data that we have. And so together, we're excited that this type of approach can actually lead us to new biology. To give you a sense for what, how this actually looked like in our study, we performed a screen with about 200 different metabolites with 3 different antibiotics shown on the left-hand side that target very specific and very different parts of bacterial cell physiology. And what you can see from the graph is that, the 3 different classes of antibiotics each elicit different types of responses to different metabolites. And you can see that by the different colors in a different column, that the columns are different from each other. But hopefully one of the things that you can appreciate is that several metabolites are also able to elicit similar qualitative behaviors across our different antibiotics. And you can see that by looking across different rows here in these heat maps, that several of the rows have similar colors, of although different magnitude of shading. This kind of informs us that perhaps that in addition to the very kind of canonical or conventional understandings that antibiotics are killing bacteria by hitting their targets, that there might also be features in bacterial physiology like metabolism that can contribute to what makes an antibiotic work well or not. And so with our overall approach, we first measured these we first took these empirical measurements. Then again, we use these metabolic models to simulate the metabolic conditions corresponding to each of these 200 different conditions. And we use machine learning to identify reactions and pathways that can explain the data that we have here. And what emerged from this analysis was several different pathways which the computers are predicting to be commonly involved in how antibiotics kill across these three very different classes of antibiotics. And one of the things that was really exciting is if you look at that top cluster, that this approach from a completely unbiased perspective was able to return to us things that we already knew about, for instance, several aspects of central metabolism like TCA cycle. But what we were really excited about was the opportunity for these approaches to lead us to new biology. And you can see that in the middle set where you see the sulfate assimilation cysteine biosynthesis, the purine pyrimidine biosynthesis, that block of pathways. And two things that were really interesting about this is that number one, these were new pathways that had not been previously implicated. And number two, that you can see from the shading that because we had the simulations available for us, we were able to use some of that quantitative data to compute qualitative pathway scores. And you can see from the shading that ampicillin and ciprofloxacin, two of our antibiotics in this case, had a different shading than gentamicin, which gave us a qualitative interpretation that these pathways would behave differently with ampicillin and ciprofloxacin than they would with gentamicin. So we had additional resolution into the biological phenomena. And so what we did was from these analyses, we started trying to understand maybe what could explain the biology that was happening in that middle block of pathways. And one of the things that we found in common when we opened up the boxes and looked at the different reactions and reaction coefficients in machine learning was that it appeared that the computer was predicting that many of these effects were explained by differences in purine biosynthesis, or differences of a very specific type of nucleotide metabolism called purine biosynthesis. So this led us to experiments back in the lab to see if indeed purine biosynthesis was at all relevant to antibiotic killing. And so we did a series of experiments where we deleted several genes in purine biosynthesis. And what you can see is that if you look at ampicillin and ciprofloxacin on the two panels on the left in comparison to gentamicin on the right, that the black line is our wild type control case. And in many of these mutants, we saw protection or decreased killing with ampicillin and ciprofloxacin, but increased killing with gentamicin. We saw similar biological phenotypes if we use a biochemical approach, in this case using a compound, 6-mercaptopurine, that inhibits the first step of purine biosynthesis. So we're quite confident that indeed that the machine learning approach is leading us to new biology that we could test and directly validate. So this led us to ask questions about why purine biosynthesis might be important for understanding how antibiotics kill. And there's several, when we turn to the literature, what's interesting is that nucleotide metabolism has been really very much well studied, and in the two classes of nucleotides, purine and pyrimidines, these two classes are directly coupled to each other through a shared substrate, PRPP, and they both possess internal negative feedback loops. And in a previous study, a former postdoc in Allumni had done some work characterizing several of these nucleotides in response to antibiotic stress, and we can see is in red that Peter Belenke had observed that following antibiotic stress, the purines are depleted. So this led us to hypothesize that perhaps the reason why purine biosynthesis is important is that the cells are trying to deal with this type of nucleotide stress following antibiotic treatment. And then if we supplement these nucleotides back, that we can actually protect against antibiotic killing. And when we did that experiment, that's exactly what we saw. So when you see here in the red lines compared to the black, that addition of adenine purines actually protects these these E. coli cells against these three different classes of antibiotics. In contrast, if we use the other type of nucleotide, pyrimidines, we actually see increased killing. So this gave us two very convenient kind of biochemical tools for manipulating metabolism and reaching opposite effects on antibiotic lethality. So this led us to ask ourselves fundamental questions about understanding why nucleotide metabolism might be at all important to understanding how antibiotic killing occurs, and some of the connection back to some of the stuff that we were showing you earlier in relation to ATP being different and these metabolic states being important. And so if you go back and look in the literature from the 90s, one of the things that's really interesting is that when you do a inventory of the metabolic costs it takes to make new cells, what you find is that nucleotide metabolism as a process is actually very energetically expensive. In fact, it costs just as much energy for a cell to make new nucleotides as it does to make new amino acids, which, you think about it, that's actually, the amino acids are the things that make all the proteins that are important for almost all cell functions. So we challenge ourselves to wonder if perhaps maybe the reason why nucleotide metabolism was related to antibiotic killing had something to do with energy. And so because we had these model simulations, we could go back to the simulations and see what the computer might be predicting would be happening in cases where we supplement with adenine as a lever that protected cells against antibiotic killing, and what might be happening if we supplement with uracil as a pyrimidine lever that might increase antibiotic killing. And what the model predicted on the top left is that if we supplement with either of these two different nucleotides, that we would have opposite effects on total nucleotide biosynthesis activity, where, for instance, in red with adenine supplementation, with the purine supplementation, that total nucleotide biosynthesis decreases. On the next panel, the model further predicts as a consequence of this that the cells that are treated with adenine use less ATP. In the third panel, the model further predicts that the consequence of the cells needing less ATP is that they decrease the amount of essential carbon metabolism activity to make ATP. And then finally, in the panel on the right, that we could actually experimentally measure this by looking at changes in oxygen consumption and respiration. And so this directly led us to a biological hypothesis that we could test, which is that we added adenine back to cells with and without antibiotic treatment, that those things would decrease the respiration used by a cell that's necessary to make energy. And in fact, when we did those experiments, that's what we see. So if you look at the black line in all these cases, on the left-hand side is control. And what you see in the three panels with ampicillin, ciprofloxacin, gentamicin, that under antibiotic stress, respiration is increasing, and that reflects an increase in metabolic activity following antibiotic treatment. But in conditions when we also add adenine in red, what you see is that increase in metabolism is actually very much inhibited and decreased, meaning that supplementation with these purines is able to block or actually or absolve the cells of the need to create more energy, because their overall demand for energy is less. And conversely, the opposite is true in blue when we add a pyrimidine like uracil. And finally, when we do other experiments to understand this in more quantitative detail, so for instance, by measuring different metabolites, different energy cofactor metabolites in these different conditions, what we see in the very left-hand panel is that when we use metrics to describe the overall ATP energy balance, that addition of adenine actually decreases the need for cells to make new ATP. And in some of in the panels in the middle, other metrics for anabolic and catabolic activity, what we see is that addition of adenine decreases the anabolic activity of a cell, which is the need of a cell to synthesize new parts. But in the third panel, doesn't change the catabolic activity, which describes the ability of a cell to take in nutrients. And so together, what you see in the right-hand panel is in that phase plot is that the addition of these purines actually moves the cells away from the control condition into a metabolic state that is more dormant, because the cells both have to both make less ATP and also are decrease in their activity in synthesizing new biomass components. And so now I'm returning it to Allison.
So just to sort of wrap this up, I'm going to discuss some recent data that we're currently generating that's pretty exciting to us. And that's looking at this question, which commonly arises in response to a lot of this new information, which is, well, if metabolism truly is so important, why is this not most commonly seen as a mechanism of antibiotic resistance? Why is interfering in these core metabolic processes not one of the main mechanisms by which bacteria evolved to resist the antibiotic? And in fact, the three main mechanisms of resistance are altering the intracellular drug concentration, modifying the target specifically, or actually modifying the antibiotic itself. None of those are directly related with these key metabolic processes that Jason just spoke about. And so one of the answers to this that we think, or that we hypothesize, is that, well, we weren't really looking for these mutations. And that's because the way these experiments are typically done are very much dependent on the growth of the cells in response to the drug. And so in the lab, what we do is we evolve cells over incrementally increasing drug concentrations, which means that at the end of this evolution, when we collect the cells and we sequence them, What we have is cells that have had to both grow or both survive the antibiotic treatment and grow over time, which means they've competed with the rest of the population, ultimately dominating by the one that is the most resistant. Everything else becomes noise. If instead of adapting cells to growth over time, we did a more metabolic adaptive approach and adapted these cells to a metabolic state over time, then perhaps we could tease apart these metabolic mutations, which would give us somewhere to look now at these clinical isolates so we can find what they are and start to think about them as druggable targets. And so this is exactly what we did, where instead of incrementally increasing drug concentration, we incrementally increased the metabolic state of the cell, except instead of having continuous drug exposure, we had short bursts of antibiotic followed by drug-free growth to remove that competitive aspect of selecting for the most resistant cells. And as you can see here on the left-hand side, from the classical evolution approach, you see that the most dominant enriched biological process that emerges are those that are in response to the antibiotic, those canonical mutations of the three mechanisms I just described. But when you do this using this metabolic approach, you see that the first thing that comes up is generation of precursor metabolites and energy, followed very quickly by ATP, ADP, and purine metabolic processes, which is everything that Jason's model just drew out and explained to you. And so I just want to emphasize that neither of these approaches are any more or less clinically relevant than the other. Both of these are artificially implemented in the lab to try and tease out mutations that emerge so that we can then go look at these clinical isolates and see if anything is relevant, so we know that it's actually occurring. And so we looked at these mutations, these new metabolic mutations, compared to the classical ones, using a library of 7,000 published E. coli genomes, about half of which were clinical isolates, the other half were not. And what we saw was that these mutations were, the metabolic mutations were significantly overrepresented compared to the canonical mutations to a similar degree, or in many cases even more so than the ones that we were expecting to see. And so that was really exciting, and that's sort of where we're going to end off today. Mm-hmm. And with that, we want to thank every collaborator that we've had working on both of these, all of these projects actually, many of which come from right here at the Broad or MIT or Harvard, and others that are farther away, along with any of the funding support and all of the additional Broad members that have been instrumental in this work. And then, of course, everybody here who tuned in to listen. And then finally, if anybody is interested in learning more about anything we spoke about, Jason's paper is already published and mine will be imminently available, and we would welcome any questions.
Yeah, thank you for your attention.
All right, thanks, Allison and Jason. So yeah, we're going to open up to questions. We already have a few come in, and if you want to add yours to the list, I'll just remind everyone that if you're on Zoom, you can add them via the Q&A feature on your toolbar. And if you're on YouTube, you can add your questions in that comments field next to the stream. Let's kick it off with a question from Morgan, first to get their question in. Did you ever explore probabilistic graphic models like Bayesian networks for metabolic modeling?
Yeah, that's a fantastic question. So in this specific instance, we did not. Part of the reason for that is that there already exists some very high fidelity and fairly comprehensive models of E. coli metabolism that are just purely based on stoichiometry. And so a more sophisticated approach, for instance, using probabilistic modeling, could be very helpful, and in fact probably would lead us to some additional new biology that we did not yet uncover. But one of the things that we did here was to see if we could pilot this approach using some tools that were much more easily accessible. And hopefully one of the things that we were able to convince you guys here is that using this more simple approach, using these purely stoichiometry-based network models, that we were still able to lead us to biology that we could actually test and validate.
Okay. Let me ask, there's another question here. This is from an anonymous listener. What are your comments on using quantified metabolite compared to RNA-seq or metatranscriptomics to look at the mechanisms of antibiotics?
I think that another really good question. Both of these, or all of these approaches actually, are widely used already and provide different insights, different types of information, all of which eventually, ideally, will be integrated together to give us a more holistic understanding of what is happening in real time. And as cells evolve, here, metabolites were, part of the metabolic network modeling. So that was the most immediate, yeah.
And to add, I think another way, that's a fantastic question. Actually, or obviously, all those data, ideally, all those different measurements ought to be compatible with each other because, we're all still measuring the same biology. And you can kind of think, for instance, the analogy of the 3 blind men touching the elephant, trying to figure out what an elephant looks like. Here, what we think is that each of these different tools will give us a different perspective on the fundamental biology. And as Allison was describing, one day we're hoping that the tools that will become available so that we can do those integrations. But right now, I think, when we're examining phenomena such as metabolism, something very proximal like metabolites is likely to be more sensitive than something more distal like gene expression.
Okay. Let me ask a question. I was curious at the beginning when you talked about the decision to switch from the kind of typical black box machine learning, who knows what's happening inside, to this white box method. Do you see, when you look at other potential uses of machine learning in this kind of biological research, other sorts of roadblocks that might need to be tweaked or figured out before you can do everything you want to do?
Yeah, that's a fantastic question. So I think one of the motivations for designing and developing the study is that we've been really challenged by some of the really fundamental challenges associated with just purely data-driven or machine learning-based approaches. Namely that, that it's oftentimes very difficult to understand or interpret what it is that machine learning is telling you when it arrives at a solution. And so in this case, we decided to develop this type of approach to apply it to antibiotics, which is something that our lab cares about. But in fact, this overall approach can be generalized to any type of biology. And I think at a high level, here again, we were using more simpler approaches. For instance, we were using a simpler type of metabolic modeling. If you read our paper, you'll find that we use a simpler type, linear regression based type of machine learning. But each of these different steps ought to be generalizable so that more sophisticated approaches can really improve the fidelity of any one of these kinds of predictions. And so we think that that this is timely. We think that this is something that can be generalized to many different types of questions, many different kinds of networks. So even more, if you think more broadly, here we were interested in metabolism and in bacteria, but maybe we could extend this exact same approach to study cancer or heart disease or other sorts of human disease that are really important, and maybe look at other types of networks, such as the signaling networks or the gene expression networks. And so as a framework, we think that network modeling can be really powerful for augmenting and enhancing machine learning. And there's many ways to do each of these different steps.
That's great. And that actually speaks to, we had a question here from Paceman about how applicable this kind of work is to cancer research. So it's interesting to hear you mention that in the list. We have another question here from Anne DeWitt. What are the plans to potentially translate this work into clinical or patient impact?
Yeah, so that's a really exciting question. Certainly with some of the targets or mutations that we found in this most recent work, we're currently in the process of validating them all in the lab, on the bench, in vitro. Some of them have already turned out to be positive. Hopefully we'll have a whole long list of new genes or new targets that have emerged from this, and that's what it seems like will happen. Following that, many of these are in coding sequences that presumably have biochemical inhibitors to them. And so I should say that following what we were already doing is next doing a sort of protein analysis to see what the actual functional effect of these mutations are, many of which will have a corresponding biochemical type, actual molecule, small molecule that we can use. Following that, things, of course, we're interested in is eventually looking at how we can implement this. And there's a lot of validation to do in between, but that's absolutely something many people here are very interested in doing.
Okay. Let me ask about, and I know one of you mentioned this in the presentation, these 3 antibiotics that they all kind of brought something different. Tell me about that decision to choose these as what you wanted to focus on in the study.
Sure. Well, as you guys probably know, there's many different kinds of antibiotics, and they work by targeting very different parts of bacterial physiology. Here, within the Collins lab, historically we've been doing most of our work with these 3 specific class of antibiotics. So ampicillin is a beta-lactam, which targets processes related to the ability of a cell to synthesize new cell wall. Ciprofloxacin is a fluoroquinolone. So these are antibiotics that target DNA gyrase or processes involved in DNA replication. And gentamicin, aminoglycoside, which is an antibiotic that targets protein translation. And certainly, there's a lot of understood biology about how each of these antibiotics kill in their own unique and independent way. But one of the things that we were interested here was trying to better understand antibiotic death physiology in a way that would be commonly shared across different types of antibiotics. And so we chose to work with 3 antibiotics instead of 1 because we expected that would improve our sensitivity for shared biology. And we chose these 3 antibiotics over several of the other different classes of antibiotics because these were types of antibiotics which are known to be able to kill bacteria as opposed to only prevent them from growing more. There are several other antibiotics for which these types of approaches could be extended, and we'd be very excited to do that.
Just to add on to that, for the evolution study that we did, we also did the same 3 antibiotics specifically to look for common metabolic mutations that would emerge rather than drug-specific target mutations so that we could identify those common practices or those common modes of action between the drugs.
Gotcha.
And maybe just add one last comment, in relation, for instance, to the previous question, we think that part of the advantage of this approach is this might lead us to new adjuvant drug targets that could be broadly applicable and useful for enhancing the activity of several different kinds of antibiotics, as opposed to, creating targets that would be very specific to individual antibiotics, which might draw out the drug development pipeline.
Got it. Let's grab, we have another participant question here who wants to know about how transferable this method is to other microbe species, and they're particularly interested in human gut commensal microbes.
Yeah.
So anaerobic species, which are going to be those found in the gut, at least lower down in the large intestines, is a project that is actually currently ongoing. Presumably, the mechanism should be maintained. The concepts should all be applicable. We don't know the answer to necessarily gut microbes yet, but we do know that across very broad general species, we have tested, for example, the very first study that I spoke about here, we validated in Staphylococcus, we validated in Acinetobacter. These are consistent of escape pathogens, including Klebsiella we did, which are some of the most dangerous or the WHO classified dangerous pathogens. And so from that standpoint, this is generally applicable, and we're looking at ongoing right now how to expand that.
Yeah, and to add, again, on a technical level, these approaches that we're developing, we expect them to be generalizable to any sort of biological organism. And in addition to the bacteria that Allison was describing, we've also extended this to other pathogens such as tuberculosis, and have active and ongoing collaborations to better understand how to improve the efficacy of anti-TB antibiotics. And so, we suspect and we anticipate that this would be very generalizable to your favorite bug, and we would be happy to email and chat with you guys about the work that you might be working on.
I want to raise up another question from Sabrina, because Allison, you started with some pretty bracing numbers on antibiotic resistance. And Sabrina wants to know, simplistically, based on your work, would you advise a family member taking antibiotics to avoid certain foods to increase efficacy? And I'll just add on too, what would you say to people who are just worried about antibiotic resistance in general? What are your thoughts on that? How you talk to people about that?
Yeah, well, so these are really great questions. I love this question from Sabrina. It's fascinating. In fact, so we can't necessarily comment on what foods to or to not eat. That's definitely something for a treating physician to do. But we can say that certain metabolites are currently being investigated as adjuvants. And in fact, one recent study from this lab has shown that glucose paired with specific, I think ciprofloxacin in this case, can significantly increase the efficacy of antibiotics. And so likely there are different combinations of commonly found compounds that you would be coming into contact with from what you're eating that will change the efficacy. And hopefully some of this work can help elucidate exactly how to improve our understanding so that we can guide people better. In regard to Aviva's question, these numbers scare me every time I talk about them, because it's an incredibly concerning topic, and it's a really difficult one to appreciate, especially from the patient-physician relationship, because individually it doesn't necessarily feel like it's the time to take the risk and hold off on antibiotics if there's something pressing going on. And yet at the public health level, every wrongly prescribed antibiotic, which is something close to between 30 and 50%, is contributing to this process. So I would say that even being extremely informed, I'm still well aware of all of the caveats that happen once we step outside the lab, and being mindful. And there's a lot of education that hopefully can increase everybody's understanding so that we can make decisions that will be beneficial long term.
Mm-hmm.
Okay, let's grab, we have a few more that came in. The outside wants to know, in terms of purely machine generated biologics, has there been something created that works in real life?
Yeah, so we haven't directly been working on, for instance, biologic compounds that, as therapies, but we do know that there are others in the space who have been using machine learning to conceive and design new molecules. Regina Barzilay here at MIT is a leader in machine learning, and she has several active projects in that space of de novo molecular discovery. And to our knowledge, to our anecdotal knowledge, yes, there have been compounds formed that have been observed to be efficacious in treating several different diseases, including some infectious diseases in mice. But of course, there have not yet been any clinical trials, in humans on those. So we're optimistic, but I think to date the data is supportive, but not yet complete.
Okay. Elizabeth wanted to know, can this type of approach be used to address the side effects of antibiotics during development of gut microbiota?
So yeah, another really interesting question. I think, again, this will speak to developing antibiotics that are more targeted, more specific, more directed towards either specific species or processes, that when we get to a point where we can actually do that, things like avoiding collateral damage of your microbiome will absolutely be at the forefront, because that is something that is most highly contributing. I shouldn't say most highly, but certainly very much contributing to the rise of resistance, because as soon as we treat and obliterate our natural microbiome, that allows for these resistant bugs to grow and take over. And so that is definitely something that specifically in the world of metabolism, how it relates to antibiotics, will be kept and thinking of in terms of as we develop new strategies in the future.
And I guess to add, there's also there's new innovations that are happening in the space of network models to simulate what happens to gut microbiome ecosystems, and so under different conditions. And so we envision an approach similar to what we described here, which in this case was very focused on one individual bacterial organism. Could be generally applied to exploring, understanding different effects that you have in these different microbiome consortia, or different microbiomes between different individuals. And you can envision that this might lead us also to new biology that can inform the way we can predict or even treat things that are related to to the gut.
We have a few other questions about just how different ways to apply the approach that you developed. Bing wants to know if you could see a future application in probiotics use and selection.
Yeah, so I think something actually that we haven't necessarily touched on, or actually Jason maybe just slightly touched on, is the fact that metabolic interactions in bacterial communities is a very known and established thing. And so capitalizing on that is sort of one of those areas which I would characterize as an eco-evo or ecological/evolutionary perspective on developing new antibiotics. So not necessarily a single small molecule that targets that binds to a specific target, but rather targeting more abstracted, higher level processes of bacterial communities to take advantage of these evolutionary dynamics that we know are occurring. And so certainly not yet, but eventually in the world of probiotics, taking advantage of metabolism specifically, because that is something that can be easily programmed into cells to interact with one another, is a very interesting and I would say very exciting area that is probably just now on the surface of being explored.
Okay. We have Laura Dunphy wants to say hi, Jason, and says it's creative use of biolog plates. Have you guys tried evolving resistance of biolog plates, and could doing so help you to predict longer term impacts of metabolism on antibiotic efficacy?
Sure. So maybe I can clarify for all of our viewers. So Laura's asking a technical question about how we did our screen. And in our case, in our paper, we described that we used compound libraries that were provided for us or that we purchased from a company called Biolog. And so the way that we did our metabolic screens is that we use these kind of pre-organized metabolite compound libraries to do experiments to understand which ones would increase or decrease antibiotic killing. And Laura's question is, can we or have we, used these same compound libraries to understand how resistance might be different under each of these different metabolic conditions? And in this specific case, to answer your question very specifically, Laura, no, we have not yet done those experiments. That would be something very interesting, and it would actually be very much in line with some of the questions that Allison is exploring. I think there are fundamental questions that have not yet been answered about understanding, what are the traits that constrain how resistance develops and what forms of resistance specifically develop. And approaches like the experiment that you were describing, where we use metabolic compounds as a way of changing the environmental conditions, would be really informative for that. And so, yeah, that's a fantastic suggestion.
All right. Thanks for explaining that question too, for some of us here who may not be as familiar with that stuff. Let me ask one last question to close us out. We've had a lot of great questions today about just applications of this kind of work. And I was wondering if there's anything else you guys wanted to add about how machine learning can be used, other works you see going on out there now that get you really excited, or future applications that make you really excited about where this kind of work could go.
Mm-hmm. Well, maybe I can share. So I think that, like I said, part of the inspiration for this work was to address these really fundamental questions about how we can make machine learning accessible to us, so we can understand what a computer is doing when it's doing its making associations or building the predictive models. And so, one of the things that I'm optimistic for is that newer approaches will come online that will really help us augment just the way that humans, as scientists, we already approach and tackle the scientific method. I'd refer some of our reviewers to a really nice book by Judea Pearl called The Book of Why. And what Judea Pearl proposes is that we can use logic type of analyses like counterfactual analysis to help us identify more systematically new mechanisms that can explain a phenomena. And I think this is an exciting area where machine learning can actually augment and accelerate by systematizing and kind of increasing the throughput of those types of activities.
Yeah, I sort of touched on this before, but I think, at least to me, the most exciting aspect of where the field of antibiotics can be going is this idea of next generation antimicrobials, which take advantage of the evolution and take it sort of into our own hands, because we've been one step behind these pathogens every step of the way. And being able to implement more quantitative approaches so that we can predict the evolutionary trajectory of bacterial populations exposed in many different environments to many different types of drugs, knowing that a priori whether we can develop these new strategies that leverage all of this information and sort of beat them at their own game.
Okay. Well, Jason, Allison, I want to thank you so much for joining us today. And on behalf of the Alumni Association, thank all of you for tuning in to today's faculty forum. The alumni staff will be sure to forward any questions that we didn't address to Jason and Allison, or if you think of something later, you can send follow-up questions or just feedback to alumnilearn@mit.edu. Thanks, everybody.
All right, thank you.
Thank you.
Thanks for joining us. And for more information on how to connect with the MIT Alumni Association, please visit our website.